My name is Ron Thibert. I'm a pediatric neurologist slash epileptologist at Mass General, and we've had a Dup15q clinic since 2010. So, again, like I said, lucky enough to be involved with the Alliance for probably closer to 15 years now. So, I think that's about it.
Okay, awesome. So for this, I don't know that we need everybody on the panel to answer each question. We'll just kind of have you guys raise your hand, take a stab at each one as we go through, and then, but a discussion is good too if we think something needs a little more time. So let's just start with the basics. What is a clinical trial? So I don't think we started that far back in our previous session. So what is a clinical trial?
Okay, clinical trial is one kind of clinical study. Usually when we call it a clinical trial, that means it's an interventional trial, meaning that you are testing an intervention. That could be a drug, that could be a behavioral intervention of some sort in people. And often we're talking about doing this in a group of patients with a specific disorder. Yes, Shafali, do you have something to add to that definition?
I don't, actually. I mean, there's different, and I think you did a nice job earlier today talking about the different phases of trials. And, we always think of the classic trial as our randomized control trial that's looking at, the effectiveness of a drug or an intervention. And that's really sort of later stages. But really, anytime we're testing a therapeutic, you could, we could define that as a clinical trial.
Thank you. Okay, so I think the next one's interesting, and we definitely didn't touch on this yet, and I would kind of like maybe a couple answers for this. So how does a clinical trial come into existence? Like, what needs to happen for a pharmaceutical company to be interested in a disease, to take it kind of all the way through from interest to development and through to a clinical trial? I think I've heard varying opinions from the community, and I'd really kind of just like for this panel to kind of discuss that a little bit.
Oh, sure. Yeah, so I could take that. I mean, I do want to step one back and say from a researcher perspective, when I think of a clinical trial in the state of which our company is working, I think of it as the goal. It's at least, if you can get to that point, that's a really, really important point, because it's not easy to get there. And then in terms of it coming into development, there are conversations that happen with regulatory agencies to discuss the timing of the candidate drug and what the trial is going to look like. There's a lot of effort into thinking about how you're going to measure the effects of drugs, how are you going to control for whether the drug is working or not working. And so I think that you start to know later in the drug process that this is going to be coming on, but I would say that that's a bit about how it comes about.
Okay, yeah. This will be a bit general, but honestly there are two things that have to come together. One is that there has to be a disorder with a clear unmet need by someone's definition, and there also has to be science and research that indicates a way to address that need. So I said that would be very general. More specifically, whatever that disorder is, of course, has to fall within an area that a group feels they can address successfully, right? Where there's some experience in that area, where one understands the population, one understands the disorder well enough to know what to look for, to know who to include in a study, to be able to design that study and to be able to find the people to take part in the study. So there is a bit of a perfect storm element to this, and I think that some of the elements of that perfect storm have been addressed in different sessions today, and that's actually given quite a nice picture of all the things that do have to come together for a trial to happen.
Yeah?
I think Dr. Thibert may have had one thing to say.
Oh, yeah, of course, sorry.
I forgot he was on the screen for a second, so I want to make sure you get to him.
Dr. Thibert?
No, I didn't.
You're okay? Okay. Well, maybe we can have you answer the next one. Why should somebody have their loved one with Dup15q participate in a clinical trial? And what might a disease-modifying outcome look like, or a positive outcome? We may just think kind of in terms of really finite improvements, like I take a seizure medication and it ameliorates seizures. But if we're looking at something kind of more broad, like a disease-modifying therapeutic, what kind of outcomes might somebody expect from that?
Yeah, we had a good discussion about this in our fishbowl. So if you were in that, then sorry for a bit of the repeat, although we'll I think have different perspectives here. I would say the key sort of point that I would emphasize around that question is that fundamentally it's always up to you as a caregiver, if it's the right thing for your family to be in a trial. There's no right answer to that outside of what your decision is based on feasibility and fit and all those things. So you should never feel forced to be or coerced into being in a trial. It is your choice in the end. But these trials this is the goal. We all work so hard, as clinicians, as researchers, to understand, and we'll be specific here, understand Dup15q syndrome, think about the mechanisms that underlie the challenges that our kids and adults with Dup15q face. All that work is to improve quality of life, is to improve well-being, to improve, various neurodevelopmental outcomes, medical conditions, and we want to make lives better. That's the goal. And how do we do that? With treatment. And how do we get treatments into the clinic is with trials. We really cannot use medications or even behavioral interventions. I mean, we use those things sometimes kind of without the kind of rigorous evidence. But the problem with that is then we aren't really sure if it's going to work or not, right? The way we can assuredly use treatments is to have them already have been rigorously tested in clinical trials. And most of the drugs that we use in other conditions for other indications, even a lot of the antiepileptics you all use, I mean, those have all been tested more broadly in clinical trials to be able to then get them approved, the medications approved. And so, clinical trials gets us to a place of being able to treat the challenges. In terms of the specific outcomes, I mean, this is where actually, many of you probably have been involved with this work, but a lot of the work we've been doing in our research on understanding the clinical profile of Dup15q syndrome, trying to understand which measures work better in our kids, which developmental measures, cognitive measures, measures of adaptive skills, which ones are giving us some signal, meaning showing us the variability in ability in our kids. All that work, and then all the data that are in ladder, which also contribute to that, all those data have helped us really understand, well, what would be the right outcomes that we would measure in a trial. And what we want to measure and what we want to improve kind of align, right? It's things like daily living skills, it's overall development and cognitive ability, it's language, it's social skills. Of course, epilepsy is a big, important target as well. It's not the focus of this upcoming trial, but it's certainly an area that we'd like to also improve. But fundamentally, the goal is to improve quality of life.
This is one I could probably weigh in on. One of the questions so far that I actually could. So it comes up a lot. The Angelman trials started a little bit before the Dup15 are about to start, so we've gotten these questions a lot, and people always ask, what do we expect to see? What what will we see? And it's tough because these first in human trials, the treatments have been tried in mice, so we're not going to put, actual human children on like a rod and see if they fall off or put them in a water maze. So it's not quite clear what's going to happen, but, we kind of expect the more kind of basic functions to hopefully be the ones that are improved first. So kind of motor skills, balance, that kind of thing. If it works even better, then hopefully we'd see some improvements in, seizures, behaviors. And then if it works really well, then hopefully, cognitive improvements, that kind of thing. But, as far as what exactly will get better, it's hard to know until we try. But these compounds don't kind of get to clinics and get to us, get to our Dup15 clinics, unless we all are kind of on the same page that there's more potential benefit than risk, and that's kind of where we're at. And as far as specific, people's specific situations, it's really important to just ask your doctor, do you think this is something you'd benefit from? Is there more potential risk? Would we be a good candidate? And then, they can kind of go through all the pluses and minuses. And then, because, some families, I think, if they're doing great, then maybe it's, you don't want to rock the boat. If you're struggling more, it might be a little bit more motivation to try it. But like Shafali said, it's everyone, everyone's kind of got to make their own decision. But, you know, we wouldn't actually have these trials in the clinics if we didn't think there was more potential for benefit than risk. And then, you know, I think as far as each individual family, it's good to just, you know, talk to your doctors and, you know, try to make that decision.
Yeah, of course.
And I just to add one thing that also actually came up, and you, Jenny, brought this up in our fishbowl, but I think it's a really important point, which is, you know, we have a very rigorous and careful process of informed consent. And in that informed consent, we provide excruciating detail about all aspects of the trial. And it's a lot, you know, but the goal is that, you know, you as caregivers, and if you decide to enroll in a trial, you're going in eyes wide open. So you really understand what the trial entails. And again, you always have a choice to you know, stay in a trial or not. But, you know, that early process of really being, again, informed consumers and asking all the questions. No question is too, you know, simple or obvious or anything, because it's not. You know, there's a lot of details that go into thinking about these protocols. And so it's really important for you all to ask the questions that will help drive your decision as to whether you want to be in a trial or not.
Yeah, so I think that's a good segue to the next question. So that's making sure that patients and families understand, now that it's in clinical trials, that, you know, you understand what it is you're taking. But what goes on behind the scenes before that to make sure that this drug is safe for trials?
Okay, so understanding the safety of a drug starts long before a clinical trial, even a phase 1 early clinical trial happens. Usually the first tests that are done just on individual cells, are done using data banks and computers to understand where there could be issues and try to foresee those. So it starts long ago. All of that information done on a molecule that is going to be given to a human being has to be made available to health authorities before that molecule, that drug, can be given to a human being. That is really important. I wouldn't have it any other way. I think we can all agree on that information should be looked at carefully by as many people who understand the data well as possible, and from different agencies, not just from the people who came up with the idea of doing a trial. So those data have been looked at before a trial is brought to you with the idea of allowing you to sign up for it potentially. The safety for a drug going into, for instance, a phase 2 trial has been looked at in a phase 1 trial, usually in healthy volunteers, but sometimes in patients specifically. During a trial, safety will be looked at. So if there's anything that's been seen where authorities or the people doing the trial are not quite sure if there might be a risk for something, that will be looked at very specifically during a trial to track, for instance, if there's a blood value that one can track to see if there's a change that would be concerning, that will be looked at specifically during that trial. So I'm speaking about this very generally, but those things should all be in place before anyone even has an option to join. To me, another important safeguard, when you're thinking about a trial in terms of safety, is talking really clearly and asking all the questions that you have with your study doctor, like Shafali, because for you individually, you need to understand what is the benefit and the risk that you can think of in a study. Some studies don't necessarily offer a potential benefit. Those are usually the very early studies where a drug is going into a human for the first time, and people do it just to contribute to science. They'll take part. But understanding for a specific trial what is the potential benefit and what is the potential risk is extremely important. So use all the sources you can to understand that, and your study doctor is a good source of expertise on that.
Yeah. So I can comment on sort of this exact thing in terms of this is what we think about all the time at this point. You know, the stuff that we work on is way past the cells. That's been happening for quite a while, and now it's more thinking about how to make this as safe as possible and assess lots of different candidates and how they are in terms of relation to each other, safe or less safe. So with the goal of being the safest and most well-vetted candidate is the one that would move into discussions for moving into clinical trials. So a lot of thinking goes into that. And it's not just starting from scratch, by the way. There are people who have been paving these paths, and it depends on the type of drug as well. And you want to design your studies to look at the effects that might be specific to that type of drug versus another type of drug.
So let's talk a little bit about like trial design. So when we think about a trial, and I know we have the current trial top of mind, and we've gotten a lot of specific questions about that, but generally, you know, who decides who can participate in a trial, and who generally can participate? So like, who's determining those factors? I feel like we keep going to you, but maybe Shafali or Ron.
Yeah, I mean, fundamentally, it's the sponsor, you know, the pharma company that decides on inclusion exclusion, but that's based on a lot of other information, right? It's based on information about safety. It's information about what kinds of endpoints we're measuring and which endpoints can we measure based on, you know, who we're seeing in the trial. There's a lot of factors that go into it, so it's not an arbitrary decision. I will say this kind of, again, big picture in the neurodevelopmental space is that, you know, the traditional model always was you had to do a trial in adults first, and then you tested it in kids because, you know, you wanted to make sure it was safe in adults, and then you could go on to kids. And the good news is that that model has changed now, because it doesn't actually make a lot of sense to do a drug trial in a neurodevelopmental condition in adults first with that just for the goal of then being able to test it in kids. We want to test, if the goal is to test kids, we'd like to, and have the trial available in kids, we'd like to actually start that as soon as possible, right? And so now we're seeing that pharmaceutical companies are willing and able to jump right in and start trials in kids. And I think this trial is a good example. You know, our age range is 2 to 11, which is, you know, may not have been the case 10 years ago. So I think that's like a really important and exciting progression that's happened. And that's, again, based on experience and people really being able to do the right kinds of safety analyses early on to be able to feel comfortable doing, you know, having trials available for kids.
Yeah. Oh, and also, you know, like, you know, for these, you know, syndrome specific, you know, kind of, you know, specific gene therapy trials, you know, the people from the, you know, from the drug companies and the sponsors, you know, they talk to a lot of us that, you know the syndrome well before it goes. So they're not just kind of guessing or just looking at papers to kind of figure out what to, kind of what to assess, what to measure. You know, they do get input from us as well ahead of time, which I think is really helpful. You know, talking to people that actually, you know, treat the kids and adults that will be part of the trial.
Yeah. I realized something that hasn't come up is, for instance, we worked with a family council, which we like to do to understand, both to fill out, flesh out what we understand about where the need is exactly, but also to understand, once we have an idea of how we think a trial might work well, to understand how we can tweak that to make it more, let's say, to make it less of a burden for those participating, because we know that every study is an effort. And so it means a lot to us to hear back from people who are similar to those who might participate. Which part of what you're telling me that I will have to do is the most burdensome? Which part makes me think this is really going to be awful for our family specifically? And then we try and go back and do what we can to make the study more acceptable.
Okay, so in terms of continuing on the idea of trial design, some trials have what's called a placebo group. So I think it's probably, we could start by saying, what is a placebo group, define what that means, and then why do you need one? Why might a trial require one and think that it's important?
You want me to tackle it, or you want to? Yeah, okay, I'll start and then she can jump in. So this is probably the most common question that I get from families when they're thinking about trials in general in my space of sort of neurodevelopmental conditions and neurology. And it's a great question, right? Because why would you want to put all that effort into being in a trial if your child is not getting the treatment. So a placebo provides us with an opportunity for a comparison group. And a placebo traditionally is sort of a fake treatment. So it's you think you're getting the treatment, but it's not actually the drug or the treatment that you're testing. So why do we need that? Well, it turns out that really the best way, and again, there are now new creative ways to sort of test other types of comparisons, but really the best way to know if a drug is working is to compare it to a group that is not receiving the drug. And only by doing so and testing the efficacy of a drug can we then take that drug to the next step, which is potentially getting it approved so that it is available to everyone, including those that might have been in the trial that didn't get the drug. So I look at it as the trial allows us to answer the question as to whether this therapeutic could help the whole community. And if we can answer that question efficiently and most effectively, which is in usually a placebo-controlled design, then that drug is available to everyone. Right? But the only way we can get there is by doing the right design early on. But it can be challenging, I know, for families to sort of come to acceptance of the possibility that their child might not be receiving the actual treatment. But that's sort of the discussion that we often have. And I will say in these trials, like we're all blinded to what the child is getting. And so we don't know who's receiving the placebo versus the drugs. We want it to be as, equitable as possible in terms of the assessments and everything else that we're doing. One little caveat I will add is that there is a pretty big literature now on the fact that there may be some therapeutic value to just being in a trial because of the fact that your child is being monitored, and that you have expert clinicians who are, and this is not specific to Dup15q at all, I'm speaking more broadly, that there may be therapeutic value in having an expert clinician monitor your child, do assessments, give feedback on the assessments, just keep close tabs. And it actually leads to this phenomenon that we talk about as the placebo effect, where kids even in the placebos get better in a lot of studies because there may be some therapeutic value in the surveillance that accompanies a trial. And again, so I say that not to say that if you're in the placebo group, you're definitely going to get better, rather that there may be some inherent value in participation, just by all the other extra sort of elements that get added to the experience outside of the drug itself.
I think that was pretty comprehensive.
Okay. I was looking to see if we're on anything. Okay, and then what do you think is the role of the alliance in a clinical trial?
Well, I think I've said this a couple times today, so I'll sound a little bit repetitive. But when I think about my experience trying to set up a trial, we wouldn't be where we are today doing trials, and I think this is true probably for every trial that goes into specifically this population, without the invaluable information and advice of the Alliance. So we talked about the databases, we talked about things that have been mentioned are the research that the Alliance has funded, all of these things that were not done specifically for this trial. They were done, and these things have to be done before you know that it's time to set up a trial. The Alliance has helped us get in touch with the right clinicians, the right clinic sites, has helped us get in touch with families. I mentioned the Family Council, understand who may be in a position to share their experience, so that we can understand what's needed, so that we can understand what people are looking for. Let's see, also you have connections globally, and this is a global trial, so has helped us get in touch with other patient advocacy organizations in other parts of the world. They've really acted as a hub for us to be able to find the information that we need to understand how to do a successful trial in this disorder, in this population, with this community.
I'll just reiterate what Eugenie said in terms of, I was thinking hub as you're talking actually, because that's very much how I think about it. And Ron mentioned that companies and companies and biotechs come to them for advice and guidance in terms of dividing or deciding on outcomes for clinical trials. And many times that direction and the network is what dictates that. You go to the alliance, and the alliance can put you in touch with the right person if you can't find who that person is. And then the accumulated knowledge and experience and data that comes with that is extremely invaluable.
To say something, so I totally agree with them. I'll just add one other thing, which is that when I think of the alliance, I don't just think of the alliance leadership. The alliance is all of you, right? The alliance is the families. And so you all really, I mean, definitely this trial today would not be happening without the alliance, because all of you contributed with, helping us, see your kids to figure out the endpoints and the biomarker and all those things, and then messaging to the community about what, everything we've been doing in terms of getting the trial ready. I think that though, moving forward, in terms of if you, driving kind of the agenda for the next 5 to 10 years, again, you all can do, it's your voices that are going to be heard in terms of what are the big areas that you think are most important that need therapeutic discovery. What are the areas in which we should be spending our time doing the research in, right? That agenda and those priorities are driven by all of you, the families. We kind of service that, right? Like because our goals as clinicians, I mean, my goal is to make the lives of every one of my patients as whole and fulfilled and happy as possible, and I think that we learn from all of you from that standpoint. And so, I have seen firsthand in other especially rare conditions where the families banded together and they fundraised and they drove research initiatives through being able to fund, young researchers like James, who's now working in a pharmaceutical company that's developing therapeutics for Dup15q, right? They funded fellowships, they funded research programs that literally were the base and the stimulus behind, eventually therapeutics, which is where we'd love to always get to. And so I think the alliance is like the key driver here. I mean, you guys are the engine. So I think that's really important, not to put pressure on all of you, but rather to kind of hopefully empower all of you to realize that, we're here to sort of really make sure that we hear your voices. And then the other thing I'll add is that I also think the alliance plays a really important role in that piece of communication. So kind of being the liaison between families and clinicians and researchers. So if there's questions that are coming up in the community, there's things that are not clear, maybe about a trial or about, certain research agendas, or there's questions that are had, they can come to us, meaning the alliance leadership, and say, could you do a webinar on this topic? Or could could we have a panel so that you can answer some questions? There's a lot of questions in the community, and we can also then service the families that way.
Okay. That actually is the kind of scripted questions that I had at the moment. I see Carrie's walking around already with a microphone. We have, I think, a decent amount of time for questions, at least 15 minutes or so. So if anybody has any questions, I know we got to a bunch. Our fishbowl was a really active fishbowl, but if there's any continuation of questions, we can take them up here too. There we go.
I was going to add too, I know some of this is repetitive, but we know people don't go to all the sessions, and this trial is so important. So thank you to our panel for being repetitive today, because I want to make sure we hear this over and over to get the message out. So thank you.
I have several questions, but I'll just do one, and then we can come back as time allows. The first question that I have is, as this was developed, we looked at mouse models to see, how this might work. How does activity in a mouse model correlate to humans, like in other trials? Like, is it a good sign if it correlates in it works in mice, then yeah, it's probably going to work in humans? Or how hopeful is that correlation?
Sure. That's a really great question. I think that how translatable, Ron made the point about the rotarod or testing, an animal walking on a rod, and how you need to measure something in the animal, but the translatability of the animal is very linked to the disorder. So I'd say that some are much more translatable than others. Dup15q has the challenge in the sense there are many, as we've been learning about all day, and during the science conference, this slide was shown every single talk, is there's many genes that are involved in the region, and showing that in a mouse is much more difficult. But you can make attempts, and sort of certainly having that information, if it's promising looking, is important to know. If it's not there, it doesn't mean that it can't be something you could find a different way to test. We routinely use human cellular models. There's equal limitations to that. There's not behavior. You're not rescuing an actual symptom. But there are correlates potentially in addition. If you can do both, that would be even more promising. So I think this gets to the hard part, which is you have to not just show the safety, but you also have to be accumulating evidence all the time that shows that your concept, I'll talk about this tomorrow, but for us it's targeting UB3A gene and reducing that. We have to show that that is actually beneficial in some way, because there are other genes there that could be important. So how do you show that? And we think about that all the time. So I think it doesn't only have to be a mouse. To reiterate again, the mouse model will depend on the disease, and some it may be more simple, but there are lots of other tools available these days to be testing these things, and as many models you can test, the better the data package and more convincing it would be.
Good answer.
That's a great
They
answer.
said that's a good answer. That was good.
Yeah.
Any other from the panelists? Answers to that? Okay. Other questions? Yes. Oh, I'm so sorry. Yes, that side of the room, and then you're next.
Hi, so I was wondering, two questions, is what are you going to be looking for in order to advance from phase 2 to phase 3 potentially? And then furthermore, what would that timeline look like if you have an idea of when phase 2 of Quindecim would end to when potentially a phase 3 would open?
Okay, for timelines, I honestly don't know, because how long a trial takes depends on how fast it enrolls. And this is a pioneering trial in a pioneering population, so it's really hard to predict. I just honestly don't know. I can say that in general, if a phase 2 trial is successful, and remember, a trial is an experiment. The reason we're doing it is because we don't know the answer. If the results are encouraging, usually, this is just kind of looking at all of pharma, what I'm aware of, a phase 3 trial can start about a year after the data are understood. And when I say after the data are understood, that's not quite the same as the day the last person finishes participating in a trial, right? So I'm just trying to give that sense of the timelines. Whether a phase 2 trial is successful depends on a number of things in a rare disorder where there's not a lot of trial experience. So one is looking at that primary endpoint, which is the main thing that you're setting out to study. However, when one is working, we talked about how you translate results, right? We've talked about understanding what typical development looks like in a specific population, right? Understanding what a child who is doing X, Y, and Z one year, what they might look like the next year. Understanding all of those things is necessary to decide which is exactly the right thing to be your primary endpoint. So where I'm going with this is to say that all of the secondary endpoints, things that are also being looked at, are also important in getting a full picture of what effect is seen in those who, ultimately in those who turn out to have been on drug and those who turn out to have not been on drug. So it will be a comprehensive look at everything that's learned in the study.
Any other answers from the panelists about that? Anything else to add? Dr. Thibert, anything from you on there? Just raise your hand if we leave you out. Okay, great.
I was just wondering if you guys could disclose the percentage of participants that would be a placebo versus participants that are actually going to get the drug that's tested, if you're able to disclose that or not.
Yes, that's a great question. The ratio in this, what we call the randomization ratio, so how many people end up on active drug versus fake drug, placebo, is 2 to 1 in this study. Often it's 1 to 1, 50% on each. We are trying to maximize the number of people who take part in the trial that do get the chance to be on active treatment. So out of 90, 60 individuals will get active treatment and 30 individuals will get placebo.
Anyone else? Any other questions? Yell if we missed you. Oh, yes, Kelly. Kelly has a few more. She's actually just a decoy here.
Yeah, I'm a plant. What else does the GABA alpha 5 receptor affect? So if we're looking at changing it, what are some of the potential things that we might also see as an effect of that?
So do you mean specifically, like if we're actually trying to modulate it with a drug, like what else might be? It's a great question, and we don't entirely know, and it might, and partly that's because, you know, our measures of behavior are very downstream of like the underlying core mechanism, right? I mean, there's definitely a lot of work that, and James sort of alluded to it, because there's so many genes in that 15q region. You know, we know UB3 is very important. I mean, it's important even for GABA signaling, right? Like and we think that probably it's like UB3A plus its interaction with with the other, you know, genes in that region, including the GABA receptor cluster that do impact, you know, synaptic sort of function, you know, brain network development, all those things probably, of course, affect, you know, brain excitation inhibition, which then, you know, sort of begets seizures and other things. So there's a lot, they have such ubiquitous effects. Isolating what the exact, like that, the one GABA receptor, what that does to a specific behavior, we don't know. And I don't think that's something we ever will perfectly disentangle. And so what we do know is we think that biomarker, which I'm going to talk about tomorrow, is a readout of somehow there being what seems like almost excessive GABA neurotransmission. But even that, I'll be honest, is a little bit of a puzzle, if you will. I mean, the biomarker tells us that's probably what's happening, because the biomarker looks like this EEG signature that all of these kiddos have. It looks like they're being treated with an exogenous GABAergic medication like a benzodiazepine. I mean, it's what, if one of us took Ativan and had an EEG hooked up, that's what our EEG would look like. So it's suggesting that there's an effect on GABA signaling. Why that happens? Because of you know, we have 3 GABA receptor subunits that are overexpressed. Why does that lead to more GABA, you know, in the synapse? We don't actually know exactly what the reason for that is and why we see that signature. But you know, to answer your question about what it might affect, we are, because of the fact that we're not exactly sure, but we know what we would like to see it affect, we're measuring a lot of aspects of development for the trial. And really that measure of adaptive functioning, which is the primary outcome, that's born out of for two reasons, really. One is that we have a lot of nice data on that measure in kids. Many of you have probably filled that measure out, the Vineland, because you were in our studies. We came to many family conferences and collected, you know, the Vineland. It turns out the Vineland gives us a lot of rich information about kids with Dup15q syndrome and a really nice range of scores. So it's not like every kid scores exactly the same on the Vineland. We see a big range. And so we think that actually is helpful as a good kind of measure of just level of functioning, if you will. And really the hypothesis is that if you can say what really gets impacted is, I'll just make this up, like motor skills, right, or language. Well, that's going to have a downstream effect on adaptive skills too. It's going to affect the way a child lives in their world. And so we like that measure because we think not only do we have data on it, but we really do think that it's an excellent downstream measure of maybe something more narrow that actually is being affected.
Thank you.
Yeah.
Thank you.
That was such a comprehensive answer. I'm trying to see if there are any chinks that I can fill up. I think just one thing to point out is, I was thinking through GABA alpha 5 receptors, and they're so hyper specific. This is such a, it's kind of, we said kind of the opposite of a benzo, but really a hyper specific partial benzo. And then I came back to, something I've had to face quite a bit, is the idea that what these receptors do and how they're affected in an individual without Dup15q syndrome will be different than what happens in a child or an adult with a brain that has been in a different GABA environment its whole life, right? So this study will just tell us really a lot. That's why we're also, as Shafali said, measuring kind of a broad set of behaviors to not miss anything that we may not be able to predict exactly at this point.
Let's take one more over here, and then we'll close up if that's okay.
Hi, good afternoon. Thank you. I have a question. What would life look like if we participated in the trial? Meaning, how many times would we have to travel? My daughter does have behaviors when it comes to going to the doctors. Like she screamed for a half hour on the top of her lungs to have an EEG. So, and I would love to participate. I don't know if that would exclude us, and like drawing blood. So that's difficult for her as well. So I'm just curious, if we did participate, what would life look like? How many times would we have to come see you? That's all, just trying to get a picture of what it would look like.
Okay. I'll start, and then maybe Shafali can say a little bit of something about what a day looks like for a family visiting her site for a study like this. And I'll just I will list a few of the burdens of the trial. Okay, so there are 10 study visits total. 4 of those at most sites will be able to be done as home visits where a nurse comes to your house. Many of the visits do include a blood draw. Okay, not easy for any child, and you know your own child best, so you know what that's like. Many of the visits include an EEG. Okay, the setup is a wireless EEG. So they'll have have to go through getting the electrodes on. Those will then be wrapped. Some sites are using a cap. Some sites are using individual electrodes, but they don't have to be hooked up to something, right? They can move around the room. They can move around the hallway, however the site is set up to enable that for you.
With the backpack or no?
Only no backpack, as far as I know. Okay, so I said I'd kind of go into the horrors. I think those are the things that for the child are probably, it depends on the individual child. You know your own child best. I think something that can be difficult for families in addition to travel is what we call direct assessments, things like the Mullen Scales of Early Learning, where your child interacts with a tester who's extremely experienced. We're only doing this at sites that have a lot of experience working with kids with neurodevelopmental disabilities specifically. So you should not be seeing anybody who doesn't understand how to approach most children. But I know that for parents, it can be unpleasant to watch your child being tested on things that you may have seen them tested on before. And you may already know that's a skill that isn't probably going to work out today. Or seeing your child tested on a day when you think they're not at their best. And you know that, if we did this a week from now, they would perform differently, and you're not getting the full, someone's not getting the full potential of your child and seeing that. So I'm mentioning things that I've heard from parents might be challenging for them, so that's why I'm bringing those things up, and I'm kind of painting the most dire picture I can think of to paint here, just to be clear about some of the challenges that you may face. I think on the positive side, some people really do benefit from having that increased contact with a clinic, maybe connecting to clinicians more than you usually would, or finding that the study staff really are supportive for you, finding that you get the clearest attention to what's going on at that moment in your life. So that's to kind of alleviate the very dire picture I painted. And let me let.
Quickly, one question she asked was, like, her daughter cries a lot for EEGs or cries blood off. That would preclude you from participating.
No. We understand that we're not expecting children to like show up and say, here's my arm, please draw my blood, or anything like that. Okay, we get that. And I think Shafali can paint a clearer picture of this as well, yeah?
I mean, yeah, everything you said. I mean, I think it's a partnership, and we completely get it. You know, most kids cry when they get EEGs, and most kids cry with blood draws, and it can be stressful and tiring. But we really have thought, you know, and I can't speak for every site, but one thing about this trial that we've been really proactive in, because I was sort of actually involved, I was working directly with Roche on the trial design, and now I've switched over to the role of being a site PI. But in that early process, like, we actually proactively started meeting with the sites that were going to be the trial sites to actually have all the PIs start talking to each other around, like, what are our questions? What are we worried about? Knowing our patient population, what, you know, things like, there's a lot of, the first 2 days are the most intensive. So long EEG monitoring. At my site, we're actually admitting kids overnight to our epilepsy monitoring unit. So you don't have to come in and sit all day in a clinic room and then go home and come back. Some sites are choosing to not admit overnight and have them actually go and come back because they have, their facilities are set up better for that. So there's some variability, but all of us have really talked about, like, knowing what the protocol is, how do we make this as pleasant for families as possible? And we'll keep talking about that as we start enrolling kids. But there's no expectation that, like, everything's gonna go perfectly. We really want to work as a partnership with all of you to figure out, you know, how do we make it as comfortable and feasible for you as possible.
Thank you. I know that we're right at the edge of time. Can we just give a hand for these guys and what they're, I know you've spent the whole day with us just talking, right? So as you guys make your way down, I'm going to ask Dr. Chamberlain, Stormy Chamberlain, to make her way up. Take your water with you, please. I'm sure y'all have been talking for so long, so thank you so much. We just want to continue to stress that if you haven't heard us say this 20 times, look at here. We thought about this and it popped up.